Hello, everybody. My name is Andreas Roos. I'm a professor for translational neuroscience working for the University Hospital in Essen in Germany, but I'm also affiliated as adjunct professor at the University of Ottawa at the Children's Hospital of Eastern Ontario. I would like to thank the MDF for this very kind invitation. It's a great pleasure for me to be here in Indianapolis. It's actually my first time in the city, and I'm very much looking forward today to report about multi-omic studies toward a better understanding of biomarker research and biomarker definition in myotonic dystrophy. Okay, so why biomarkers are important? Of course, they're not only important in the context of myotonic dystrophy, they are also important in the context of other neuromuscular diseases. They enable us patient stratification. They might support actually the monitoring and the predicting of the disease progression. They may, in the best case scenario, enable us to evaluate the success of therapeutic intervention concepts. And also, that's less important for myotonic dystrophy, but of utmost importance for other neuromuscular diseases, the biomarkers allow us to evaluate the pathogenicity of variants of unknown significance. That's a very important issue now in the era of next generation sequencing. In fact, a biomarker is defined as any kind of measure helping us to distinguish between a healthy status and a disease status. And there are many different types of biomarkers. So we have imaging biomarkers, for instance, some changes in the muscle MRI, where certain muscle groups are affected in terms of a selective vulnerability, may serve as biomarkers. The presence of mutations, like in the case of DM1, so the CTG repeat expansion is indicative for the presence of the manifestation of a subtype of myotonic dystrophy type 1, and therefore counts as a genetic biomarker. And of course, also muscle pathology findings, like in some cases of DM2, the presence of vacuoles may serve as biomarkers. And also different molecules have been described as biomarkers in this context, including transcripts, but also microRNAs, metabolites, and my favorites, the proteins. In fact, we have different type of biomarkers. So we have to distinguish on one hand between diagnostic biomarkers or disease biomarkers, which are indicative for the presence of a disease, and predictive biomarkers, which may help us to predict whether a patient will respond to a therapy or not. We have, in addition, also therapeutic biomarkers, which allow us to conclude whether a drug or therapeutic intervention was successful or not. So we definitely face some complexities in the biomarker research, especially in the context of myotonic dystrophy. We are talking about a relatively rare disease, which is clinically very heterogeneous, and we also face a problem here of the somatic instability. As Darren already mentioned quite nicely, the repeat expansion in blood cells is definitely not reflecting the repeat expansion in muscle cells. So when we do some biomarker research on tissue or on cell lines, all the results have to be interpreted with caution. The muscle biopsies, which are very often used to study biomarkers in the context of tissue markers, are becoming less available in the context of genetic testing. So they are less commonly collected by neurosurgeons for diagnostic purposes, which may also limit us in the identification of tissue-related biomarkers. But nevertheless, when we also talk about tissue-related biomarkers, we have to take into consideration that the molecular information we are getting out of these tissues are only a snapshot in time. And we also may face the problem that we struggle to differentiate between primary processes, which are the drivers of the manifestation in the tissue, and secondary events, actually just as a trigger of muscle cell degeneration, for instance. So we have different sources or sources of biomaterial, which enable us biomarker research. I already mentioned that we have tissue, so muscle tissue, or also sometimes we have nerve biopsies available to study biomarkers. We have different in vitro models. Darren already mentioned the fibroblasts and the myoblasts. And we also have different body fluids, including urine samples, CSF, but of course also the serum and the plasma samples. And I'm not going into all these details with regard to the different advantages and limitations which are coming along with the different biomarker sources or biomaterials. But I want to highlight, especially in the context of proteomics, that especially tissue, muscle tissue, and serum samples are coming with some analytical problems, analytical challenges. So we have very high abundant proteins in either of these biomaterials. In the case of muscle, we have a high abundance of certain structural proteins. In the case of serum, a high abundance of albumin, what we call in the proteomic field a high background, which is in turn quenching the dynamic range of the proteins which we can monitor. So we actually face the problem to quantify low abundant proteins, which in turn may serve as very valuable biomarkers. When I prepared the presentation, I was doing some PubMed research in order to get an idea how many studies, biomarker studies, have been published for myotonic dystrophy type 1. And I was quite surprised that it is I was amazed on one hand to see it's among the top 3, but surprised that actually more biomarker studies have been described in the context for SMA than for DM1. And I want to mention that the number of publications is in fact definitely not reflecting the number of biomarkers existing for the different diseases. And this is simply related to the fact that the publications related to DM1 in one publication mention more than one biomarkers, as we will see later on, in terms of biomarker fingerprints, whereas publications related to SMA most of the times introduce one marker protein. Although many publications are out on biomarkers for myotonic dystrophy type 1 and type 2, whereas there is a clear predominance for type 1. If we compare this to other biomarker studies like here for Parkinson's disease, I guess it's quite clear that there is still a lot of space of improvement with regard to the different biomarker studies when we compare the PubMed entries. So 285 against or versus more than 10,000 publications related to Parkinson's disease. The title already mentioned that I'm also going to talk about multi-omic studies, and in fact, multi-omics is or refers to the integrated analysis of various omics layers to gain a comprehensive understanding of biological systems. And this is clearly coming along with a lot of different advantages. So it enables us to obtain information on a multi-layer level. So we get a more profound understanding of the complex biology which is driving the manifestation of a disease. And this is in turn also promoting the precision medicine. In fact, we are talking about the most prominent layers, including phenomics, so clinical data or ultrastructural findings we can very often obtain in cell lines, but also, of course, in tissue, genomic findings. So basically everything which has been covered in the talk by Darren already. Transcriptomic findings are of utmost importance in the context of myotonic dystrophy, as these are splicing diseases. Proteomics, so the impact of the tandem repeat expansion on the abundance of different proteins, metabolomics, and also epigenomics. I made a research of the different omic technologies. How often have those been applied in the context of myotonic dystrophy type 1? Because almost non-publications are out for myotonic dystrophy type 2 in this context. Clearly, there is actually a dominance of transcriptomic studies, which makes sense, take into consideration that it's a splicing disease. So 105 publications for transcriptomics versus 33 publications for proteomic profiling. Unfortunately, only 7 papers so far for metabolomic studies in the context of myotonic dystrophy and 54 papers in the context of epigenomic studies studying the methylation pattern across the entire genome. So when we look for multi-omic studies, I was quite surprised to see that there are only 3 publications. And it's a matter of debate if we really would define those as multi-omic studies because they only combine also just 2 omic technologies. So if we would say a multi-omic study is a study where at least 3 different omic technologies are applied, we have to admit there is no multi-omic study existing for myotonic dystrophy. With regard to the different biomarkers, especially with regard to tissue biomarkers or more precisely the muscle biomarkers, I guess it's also important to note that there are also different physiological biomarkers existing, like the 6-minute walking test or the 10-minute walking test or the results of these freeze test or the sit and stand test. And we will see much more of these biomarkers based on data we are collecting now with the smart watches or also with the smartphones. And that's quite important because these data allow us to intersect actually the physical appearance of a patient with proteins which we know are dysregulated in the tissue of a patient. And there is a list of different proteins which have been introduced in the context of different studies, including the downregulation of the DMPK protein itself, or upregulation of the muscle-blind protein, or also the Stauffen-1 protein, or other ribosomal proteins. And in fact, there was a very nice study published by the colleagues from Canada, by the team of Elise Duchesne, where she performed a physiotherapy study. So the patients underwent a 12 weeks protocol of exercise, and she collected muscle biopsies prior physiotherapy and 12 weeks after physiotherapy, and performed proteomic profiling, and then actually was able to define a kind of molecular fingerprint by showing that a variety of proteins are dysregulated after the exercise. And the majority of these proteins belong to metabolic activities as well as to muscle cell differentiation and myogenesis. So quite nice here to see how physiotherapy already after 12 weeks is changing the muscle proteome and shows a positive response also on the molecular level. In the context of a follow-up study she performed with the colleagues from Newcastle, they were focusing on the mitochondrial proteins by immunofluorescence studies, and could also show here, I just picked 3 paradigmatic examples, that the exercise protocol is changing the abundance of mitochondrial proteins, thus having a positive influence also on the cellular metabolic state in skeletal muscle. I was a bit surprised to see, although that FGF21 as a serum biomarker for myotonic dystrophy type 1 reflecting mitochondrial function has been introduced already, that the study did not contain serum studies in order to link the restoration of mitochondrial protein expression also with changes of FGF21 in serum as a kind of minimal invasive biomarker. Now I would like to lead over from the tissue biomarkers to biomarkers in cell lines, because I guess the cell lines are of great value when we think about preclinical studies. And here just an example based on the very few studies on myotonic dystrophy type 2. We know that myotonic dystrophy type 2 patients may present with vacuoles in the muscle biopsy, and this very nice paper also showed the presence of vacuoles in cultured fibroblasts derived from myotonic dystrophy type 2 patients. However, when we compare actually data obtained in non-dividing cells, so differentiated muscle cells, versus proliferating cells like the fibroblasts, we have to take into consideration that whereas in the differentiated cells, the majority of the CUG repeated transcripts remained within the nuclei, in dividing cells, it has been described that also a certain amount of this mutant transcript is transported to the cytoplasm. Taking this into consideration, we decided to perform a study making use of fibroblasts, focusing on GSK3 beta targets in order to determine whether these fibroblasts may serve in future also as a valuable in vitro model to screen for drugs which are targeting GSK3 beta function. And indeed, when we actually analyzed our data obtained on fibroblasts derived from patients with more than 2,000 repeat expansions, we could see that not only the GSK3 beta protein itself as a very good proof of principle is increased, but also 9 other known target proteins of GSK3 beta were significantly dysregulated in patient-derived fibroblasts. So we aimed then to check whether this is of clinical significance. We performed the in silico studies and checked for the expression of the different target proteins. I just selected here calpain-1 as a paradigmatic example, and we could see that all our candidates were expressed across the different cells or cellular populations known to be vulnerable in myotonic dystrophy type 1, and could also, based on immunostaining, here making use of cardiac tissue from the mouse model, show that this increase of the proteins can be verified in tissue derived from a mouse. So next we wondered whether this approach is also somehow allowing us as a kind of first step to identify biomarkers which we are able to monitor in a minimal invasive fashion. And we decided to go for a multi-tissue Multi-Omics or D-omic study, depends on how we want to define it. Again, we started with the fibroblasts. We filtered our data for extracellular matrix proteins, as we know that these proteins are drivers of fibrotic remodeling. One protein was clearly catching our attention, called periostin, which was significantly downregulated in fibroblasts derived from the patients compared to the controls. We verified this finding by making use of immunoblotting as an alternative analytical approach, and also could see quite nicely that with increased repeat size, the level of periostin in the cells are going down. We also proved this, and actually we could see that the downregulation within the cells is corresponding to a forced secretion of the protein by the cells. And this was also going along with an increase, as we could see on the transcript level, of known activators of periostin expression. So periostin was interesting for us because it's known that it's a driver of fibrotic remodeling, especially in the cardiac tissue. And there was a very nice study on a mouse model of delta sarcoglycanopathy where the patients the authors knocked out the periostin gene and could show that the knockout of periostin in this mouse model was stopping the fibrotic remodeling of heart and skeletal muscle. This actually prompted us to investigate whether we also see an increase of periostin in muscle biopsies derived from DM1 patients. And I guess there is no doubt, based on the immunoreactivity of periostin highlighted in red here, that periostin is enriched in the fibrotic scars in muscle biopsies derived from DM1 patients. And we could also even prove that this is the case in the cardiac tissue based on the availability of a heart biopsy derived from a DM1 autopsy case. So given that periostin has already been described in the literature before as a blood biomarker in the context of fibrotic remodeling, we wondered whether we can also see a decrease or change in periostin level in serum samples derived from myotonic dystrophy patients. To address this question, we investigated two different cohorts. So the British cohort, which was provided by Hans Lochmüller, and the Canadian cohort, which was provided by Elise Duchesne, and could clearly see a decrease of periostin in both patient populations. This decrease was quite nicely correlating with CTG repeat expansion. So that's actually quite a nice example of how we can intersect a protein biomarker with genetic data. And of course, this is also then quite nicely correlating with the age of manifestation. Given that we had quite robust data with regard to cardiac involvement for the British cohort, we could even show that the level of periostin are correlating with the presence of left ventricular peak torsion in terms of a stratification marker. So starting with fibroblasts, we were able to identify or define a minimal invasive biomarker of pathophysiological relevance. And we basically then now aim to do the same for myotonic dystrophy type 2, because we know, based on new pathology findings, some patients present with COX-negative fibers indicated for mitochondrial perturbations, also with some alterations on the NADH trichrome stain or on the Gomori stain. And when we perform proteomic profiling, making use of muscle biopsy specimen derived from patients, we could see a profound dysregulation of a variety of mitochondrial proteins indicated for metabolic or of a metabolic defect in skeletal muscle derived from DM2 patients. So taking this as a starting point, of course, we were interested also now in the definition of minimal invasive biomarkers. And given that FGF21 is a very well-known mitochondrial biomarker reflecting mitochondrial dysfunction in skeletal muscle, we studied with the team of Alexander Mensch in Halle in Germany the FGF21 level in serum samples derived from more than 50 DM2 patients and could see an increase compared to the healthy controls, not to the similar state like mitochondriopathy patients, but there is clearly a difference, and we are just currently in the process of writing up the manuscript. So I guess that was already quite a nice bridge towards liquid biopsies and how informative these liquid biopsies can be. They are minimal invasive. They enable us thus to start larger cohorts. We can go for repetitive sampling, which is very important if we aim to define biomarkers which allow us to draw conclusions regarding disease progression, or especially with regard to biomarkers which would give us an information regarding therapy response. We are closer to molecular changes. We can correlate also with the genotype, because most of the time genetics is done based on blood samples and the DNA is extracted from the white blood cells. I showed based on the two examples, we are able to identify minimal invasive biomarkers of pathophysiological relevance. And very important, these biomaterials, so serum and white blood cells, are both suitable for multi-omic studies. So in fact, there is a list already existing. This was a review which quite nicely the different biomarkers or liquid biopsy biomarkers known for myotonic dystrophy type 1 up. On the forefront, the new filament light chain, the GFAP, and the T-tau protein. There was a very nice study published by the Dutch colleagues where they investigated the transcriptomic signature in serum samples derived from DM1 patients, from the patient cohort of the recognition study. And on the baseline samples, they could also here introduce transcript or biomarker transcript signature by showing that 4 of these marker transcripts are clearly correlating also with the CTG repeat expansion and thus the genotype of the patients. And the result of the studies even showed that there were some biomarkers or transcripts serving as biomarkers correlating with a positive response of the cognitive behavioral therapy in this patient cohort. So a clear indication that the serum samples also might serve as a very valuable biomaterial for transcriptomic studies. In fact, I already mentioned the white blood cells, and there are many publications in the literature where white blood cells have been studied in the context of other neuromuscular diseases, but however, thus far, not in a systematic way for myotonic dystrophy type 1. So when we think about multi-omic studies in the setting of clinical trials, we have to, of course, we have many advantages. So we are even able to perhaps discover new targets for drug screening. We can identify biomarkers enabling us an early diagnosis or prognosis or therapy response. We get a very comprehensive understanding of the disease mechanisms and also thus of the mechanism of drugs. We can define personalized treatment concepts and have better resources for a more robust patient stratification. However, this is also coming along with multiple challenges. So repetitive sampling means we need to go for biomarkers which we can monitor in a minimal invasive fashion. So we have to collect clinical performance data in order to correlate the molecular changes with changes in or improvement in the phenotype of the patients. We have to select a biomaterial which is suitable for multi-omic studies, but we also have to consider when we think about multi-omic studies for clinical trials about very high costs. So omic studies are quite expensive, and especially lipidomic studies and metabolomic studies are by far more expensive than proteomic studies. And it's also quite difficult to standardize the different omics data across the different platforms. So that's a higher challenge than we know it from genomic data. And that's something we hardly faced. So when we go in our team, when we go for data intersection, we clearly need a bioinformatician on board who is able to intersect all these data and interpret all this very complex information. So I guess it's quite clear that there is actually only one biomaterial ticking all the boxes. So the liquid biopsies, I'm a big advocate of the liquid biopsies. Serum samples allow us to monitor circulating biomarkers, which may reflect muscle cell degeneration, like we saw it for the periostein, but also for the FGF21. Proteomics and transcriptomics can be easily performed on serum samples, but also on white blood samples. But metabolomic studies and lipidomic studies are quite challenging because we face the problem of many confounding factors in this case. So in the ideal scenario, we would combine both sources of biomaterial because circulating biomarkers in the serum, cellular biomarkers in the white blood cells. We would go for longitudinal sampling and we would correlate all this quite nice molecular information with clinical data. But again, we would need to have a bioinformatician on board to make sense of all of this. With this last slide, I would like to highlight on one topic which we are quite often facing when we, for instance, submit a paper introducing a new biomarker. We very often get the question back, yeah, it's quite nice that you have a new biomarker, but that's not a specific biomarker. There is no doubt that a specific biomarker would be great, enable us a much better patient stratification and so on. But the definition of a specific biomarker would mean that we would need to rule out a dysregulation in all other neuromuscular diseases. So in fact, actually, we would need to test with biomarkers across hundreds of new muscular diseases with a limited amount of patients available for these studies. And I guess this in turn is an advocate again for biomarker signatures we are going to define by multi-omic studies. So having that said, I would like to thank you for your attention and also all the different people.
So please find Andreas at the poster session and other times and ask questions. And as we leave, I'm going to ask you a question. But thank you, everyone, for a great talk. Thank you.